All right, good morning, everyone. The end is in sight. We are going to crack open the final theme of this course, evolving physical structure and neural control policies for autonomous machines simultaneously, evolving bodies and brains, which is what the field of evolutionary robotics was set up in the first place to do. So it's taken us a while to get here, but we're here. Before we talk about evolving and brains, let's talk a little bit about final project. Most of what I've seen from most of you looks like you're making good progress. People are being good about managing expectations, breaking ambitious projects down into reasonable weekly bite-sized chunks, revising those weekly work packages when things don't quite work out as you expect them to. All looks good. I saw a couple of you this week about the mass patch. So on the tips and tricks page, there's a bunch of things you can sort of add to PyroSim. PyroSim is, among many things, a shield that is meant to shield from you all the nitty-gritty details of the underlying physics engine, which is PyBullet. Nitty-gritty details like this. Has anybody actually looked at a URDF file or an SDF file? I think I forced you to do so in assignment 1 or 2. A point about the mass patch. PyroSim by default assigns a value of 1 as a mass to every link that you create your robot from. So if your robot is made up of 9 links, it has a mass of 9. Just a reminder that in most physics engines, including PyBullet, there are no actual units. So each link weighs 1 what? 1 kilogram, 1 pound, doesn't matter for our purposes. The mass patch challenges you to add a little bit of code to PyroSim that opens up or exposes this particular parameter to your own code so you can change the mass properties of your robots. Some of you are working on bipeds. One way to make it easier for a biped to walk is to make the feet heavier. Some of you are making a baseball robot. If your robot has the same mass as the ball that it hits, what happens when the robot hits the ball with its bat? Doesn't go very far. Or the robot goes plenty far and the object doesn't necessarily go far. So for many of you, altering mass distribution makes sense. The formatting of these URDF and SDF files is that numerical parameters like mass, for example, needs to be surrounded in quotation marks. Why is that the case? I have no idea. That's just the way PyBullet works. So for example, mass URDF.py, the part of PyroSim that writes out the URDF file or writes out the mass part of the URDF, you'll see that by default, this part of PyroSim just writes out this string, writes out a default mass of 1. So we could alter this to do the following, and then put whatever mass value you want in there. But now we've lost the double quotation marks. An easy mistake to make. I saw that a couple of you made it. You need to do something like this. Fidgeting with formatting. It's a bit of a pain, but there you go. Okay, there's a couple other places like that in the patches and the tips and tricks. If things aren't working, it's usually because you've altered PyroSim so that it no longer writes out a compliant URDF and SDF file. If you're getting that kind of error, have a look, and if you're still stuck, come and see Freya or myself and we'll get you unstuck. Okay, any other questions about things in the final project? Yes?
When you're initially starting the simulation, is there any way, so like if you have 2 cubes, is there any way to have it start like slightly offset or turn?
Yes, okay. So how do you set the initial orientation of objects in your simulation? You can, so let's see, it should be in here somewhere. Yeah, here it is. Okay. So again, we're looking directly at a URDF file, and so I am highlighting all of the information in a URDF file that describes how a link works. So here's link, open link, close link. Everything in here specifies physical properties of the link. Again, PyroSim is hiding most of these details from you. Let's see if you can read the matrix directly here. What material inside this highlighted formatting do you need to change the initial orientation of a link? Just as a reminder, remember that what the physics engine is doing for you, what PyBullet is doing, which sits underneath PyroSim, what PyBullet is doing is computing all of the forces that are acting on every link and updating the position and the 3D position and the 3D orientation of the object. But in some cases, you might want to start with a particular orientation. You can actually alter this code or have PyroSim alter how it writes out this code to set the initial orientation of a link. Where is it in here?
Is it the RPY parameter?
It is absolutely the RPY parameter. RPY stands for?
Rotating minimum?
Close. You're on the right track.
Roll, pitch, and yaw.
You roll, pitch, and yaw. That's it. You'll see that when you create a link, at the moment, the default is 0 roll, 0 pitch, 0 yaw, and I always forget which is which. This is one of them, this is one of them, and this is one of them. That's how, right? So go back and have a look at the mass patch, which allows you to expose mass to initial settings, do exactly the same thing, but for roll, pitch, and yaw. And if you figure it out, let me know and I'll add it as an orientation patch to the subreddit. Other questions?
Could you go into more detail about what you're expecting for the milestone 4's A/B testing?
Okay, sure. What we're expecting. Okay. What we're expecting in deliverable 4 is preliminary evidence of your A/B testing. What does that mean? So Freya or I are going to grade your deliverable 4. What are we looking for? We're looking for, we understand what your version A and version B is. Hopefully we probably already know what that is from your previous 3 deliverables, but make that clear. And then what we would like to see is a series of screenshots or a video demonstrating algorithm A and algorithm B in operation. Is there something that we can see that demonstrates the differences between A and B? What we are not looking for in deliverable 4 is for you to prove that A is better than B or B is better than A. That's what you're going to attempt to do during the exam period. Demonstrate to us that one is better than the other, or that there is no difference between A or B. Hopefully what doesn't happen for you in the final project is, I don't know, I did one run of A and one run of B, and they produce kind of similar behavior, but a little bit different. I don't know whether A is better than B or B is better than A, or there's no difference between them. Yeah? We're hoping that you collect enough data to present to us during the final project. And we'll talk about that next week. We'll talk about the final project oral presentations next week. But for milestone 4, can you show us that you've got A and B up and running? And the idea is between deliverable 4 and your oral presentation, I guess that's about a week's time, you're going to let this cook away on your laptop or whatever computational resources you have, and then try and prove to us A is better than B, B is better than A, or there's no difference between A or B. For some of you, it'll be relatively easy in the 4th and final milestone to show us visually that there's a difference between variant A and B, because for some of you, A is the quadruped and B is the hexapod. We can see it. It's pretty straightforward. For others of you, it's not going to be so obvious because maybe you made changes to the cognitive architecture of the robot. So what's okay in deliverable 4 is show us some screenshot and some video, and you might also show us your terminal. So print out some statements so that we've got some data, or Freya or I can see what the difference is between A or B. It's up to you to figure out how to show us that you've got A and B working, and that whatever the difference is between A and B, you make it visible for us. Make sense? Okay. Any other questions about that? Okay, so let's see, where are we now? Tuesday. Next Tuesday, a week from now, I will go through exactly what's expected for the oral presentation and the written report for the final project. Okay. All right, so as promised, this has been a long time coming. We are going to talk for the remainder of this course about expanding an evolutionary algorithm so that it can simultaneously tune the physical structure and the neural controller of your robot. Several times in this course, we brought up the difference between learning and evolution. Generally speaking, in nature and in robotics and machine learning, learning implies changes to a neural network or changes to the controller of a robot. But of course, evolution doesn't work that way. Evolution doesn't respect our boundaries. Mother Nature simultaneously and is continuously tuning and tinkering with all aspects of the phenotype of organisms. The idea, the philosophy behind evolutionary robotics is that if we want to create autonomous and safe machines, it's not enough to just come up with an idea for a physical structure and then train a neural controller for it and hope for the best. In the long run, if we want to create autonomous and intelligent machines, we need to find ways to autonomously optimize physical structure and the behavior of the machine itself. That is a particular stance that may or may not be true. I work in the field of evolutionary robotics. I happen to believe that's going to be how things are going to go in the long run. You don't necessarily need to believe me. What we're going to do in lectures 22, 23, 24, and 25 is look at how to do this. How do we expand an evolutionary algorithm to optimize brain and body simultaneously? Okay. All right. So as you can see here, we're going to look at the evolution in brains and bodies of 4 different classes of robots. Traditional rigid robots today, like the ones you're familiar with. Rigid links that are connected with rotational joints. The robots you're going to see today look very familiar. Next time in lecture 23, we're going to look at soft robots, where we no longer assume that we have rigid links connected by rotational joints. We're going to look at blobs of soft and rigid material, and we'll discuss in lecture 23 why you might want to do so. Why might you want to create or automatically design soft robots? Final week of the course, we'll look at evolving the bodies and possibly the brains of robots that are built from biological rather than technological components. These are biological robots, or more commonly now known as xenobots. And finally, in lecture 25, we'll look at arguably not an evolutionary approach, but a way to try and optimize bodies and brains of robots without having to fall back on random trial and error. Can we remove the randomness in evolutionary algorithms and design bodies and brains automatically in a more computationally efficient manner? Okay, let's start with rigid robots. As we've done several times in this class, we're going to go way back in time to the early 1990s. Here we go.
That were evolved to perform specific tasks in simulated physical environments. Swimming speed was used to determine survival. Most of the creatures are results from independent evolutions. Some developed strategies similar to those in real life. Once they're evolved, multiple copies of these creatures can be made and simulated together in the same environment. The next group of creatures were evolved for their ability to move on a simulated land environment with gravity and friction. Some simple solutions with just 2 parts were found. Some seemed like they could use some assistance, while others were fairly efficient, such as this rowing-like behavior. Here is an odd cousin of the previous. A mutation caused him to tumble. Some creatures evolved to incorporate contact sensors in their control systems. Here is another inchworm-like creature that tends to go in circles. This was actually a creature first evolved for its ability to swim in water, then later put on land and evolved further. A successful sidewinding ability resulted. Here is one with a hopping style. The protrusions on its arms seemed to help prevent it from tipping over. This was the fastest, with a successful galloping-like stride. This group was evolved for their jumping ability. This group was evolved for their ability to adaptively follow a red light source. The resulting creatures are now being interacted with. A user is moving the light source around as the creature behaves. This one seems to flail randomly, but somehow still manages to approach the light. Perhaps it is mean to move the goal away just as it arrives. Here is one that has propeller-like fins which are tilted depending on the direction of the light. It can adaptively swim up or down very well. This final group of creatures was evolved for their ability to compete for control of a green cube. The creature closest to the cube at the end of the simulation is the winner. Here, a strategy first arose for simply tumbling towards the cube. Then one learned to block out his opponent. But then later, one learned to overcome the obstacle by climbing over it. Some pinned down their opponents. Some covered the cube with protective arms. Others simply unfolded onto the cube. The success of a strategy is often highly dependent on the opponent. Here is a hockey-playing creature, which takes the cube away and wins by a large margin. Here are two similar hockey strategies battling it out, with appropriate gestures. This crab-like creature walks well, but often continues past the cube and instead seems to prefer beating up on his opponent. Against the arm, the crab seems to simply walk away. A successful strategy is this two-arm technique that swipes quickly in from the side and moves the cube over to a second arm. These are the final rounds of competition amongst the overall best. Finally, the seeker arm goes against the side swiper, but the cube is just out of reach.
We spent a little bit of time last time talking about parallels between those agents and human behavior. Same thing here, right? Why go after the cube when you can just beat up on your adversary? Okay. Observations from those videos before we dive into the details. What did we just see?
Really impressive for 1994, but robots assembled from rectilinear blocks, kind of free-form joined to each other in varying environments.
Absolutely, right? So you can see clearly the body plans are being evolved along with the neural controllers of these robots. Absolutely state-of-the-art for 1994. Well ahead of its time. Other observations from what you just saw? Two parts to the video. Obviously, in the first part, we were looking at individually evolved robots. What did we see in the second part? Aside from robots beating up on each other. Competition. Where did that competition come from? Coevolution. So we'll see towards the end of today's lecture another twist on an evolutionary algorithm, which is instead of evolving one population of agents, why don't we evolve two populations of agents where the fitness of any one individual in this population is a function not only of what it does, but what the individuals in the other population are doing when they interact with that individual. That's coevolution. Okay, let's talk about the technical details of what we saw just now. As Nate just mentioned, this was work well ahead of its time. I mentioned that physics engines weren't invented until 2000, 1994 here. This is clearly a physics engine, and a pretty sophisticated one at that. This was the work of Carl Sims, a computer graphics researcher. Carl was hired by a company called Thinking Machines back in the late 80s and early 90s. Just a little bit of computer history for you here. This was an attempt to create the first parallel supercomputers. We'll see the connection machine in a moment. This particular computer was designed to have multiple processors inside, and those processors could talk to one another. Sounds familiar? It wasn't just the software here that was ahead of its time, also the hardware on which this evolutionary robotics experiment was conducted. Thinking Machines Corporation, as they were developing this computer in the late 80s, early 90s, were thinking to themselves, well, it's an awesome computer, it's got huge compute, but what would anyone ever do with it? Why would you need to have a whole bunch of things being computed in parallel? The CEO of this company saw some of Carl Sims's early computer graphics work and some of these virtual agents and realized if you want to try and evolve populations of these things, like populations of organisms evolve in nature, wouldn't it be great if each node in this machine were dedicated to simulating one of these creatures? And if so, we could simulate a whole population of these creatures simultaneously. So you can see early on here in the early 1990s, a lot of the basic assumptions of how we now go about evolving robots in silico and then transferring them to reality were laid down in this initial experiment. Okay. All right, so the connection machine here, the particular machine that was used was the connection machine 5, which had 1024 cores, and it had a peak speed of 131 gigaflops per second back in the early 90s. This is a Mac Pro from a few years ago now when I made this slide, 91 gigaflops per second. So this big machine here had about the same compute as most of your laptops at the moment. Not bad, actually, for the early 1990s. All of the experiments that you just saw that Carl ran on the CM5, Carl used a population size of 300, so had about 300 robots being evolved in parallel and evolved it for about 100 generations. And that took about 3 hours on the CM5 at that time. So not that different probably from what you're doing right now, just on a much, much bigger hunk of metal. If you go back and watch Jurassic Park, one of the nice things about the CM5 is it had these nice flashing red lights on it. And you can see that actually in the control room of Jurassic Park in the first movie. What are the red lights representing, do you think? Aside from just a nice bell and whistle to help sell CM5s, they actually had a functional purpose. What do you think it meant when all the red lights were off or all the red lights were on?
Was it RAM?
Maybe. How many red lights do you think there are? Too many to count. 1,024. Why did you think that?
It makes sense that the lights are indicating how many cores are currently active.
Absolutely, right? So at a glance, you could tell how efficiently the machine was being used. So it was actually set up for diagnostic purposes. And some Hollywood exec passed through the connection machines offices and said, that's awesome. We need that in our new movie that we're making about dinosaurs. Okay. All right, so that's the hardware. Let's talk about the software now. We're going to talk about the genotypes and the phenotypes in Sim's experiment here. Just as a reminder, genotypes is the blueprint, right? All of the encoded information about, in our case, in all the experiments we've seen in this course, that genotype contains some information about the robot. In your assignments, your genotype probably is still a string of floating point values, which specifies the weights of the synapses inside the controller, which is inside your robot. There are many, many parameters of your robot, like, for example, the masses of the links, that are not encoded in the genotype. The phenotype is the thing that's built from the genotype, built from the blueprint. Again, in this course, the phenotype for us has usually been the geometry of the robot, how it moves, and its controller. So phenotype is sort of this umbrella term for the thing produced by the genotype. The genotype, again, can specify more or less of the phenotype. Some aspects of the phenotype is specified manually up front by us, and we turn some of those phenotypic parameters over to the genotype or encode them in the genotype and let evolution figure out what those parameters should be. Yeah? Okay, that's the genotype to phenotype. What we haven't talked about too much in this class yet is the G to P map. I think I've mentioned this in passing a few times now. The genotype to phenotype map is the algorithm or the mapping or the process or the function, the verb, the thing that transforms the genotype into the phenotype. In the case of us, biological organisms, that mapping is fantastically complicated. It's usually referred to as development. The way your genotype influences you as a single cell into the thing that's sitting here in the room with us today. Simulating that whole process in silico, nobody's figured out how to do it yet because it's fantastically complicated. Question number one, how do we do it? Question number two, should we do it? If we want to automatically design autonomous and safe machines, do we need a fantastically complicated genotype to phenotype map? Nobody knows yet, but in this experiment today, we're going to see the most complicated G to P mapping we've seen so far in this course. And you just saw it produces some pretty sophisticated results. Not bad. Okay, so let's start with the genotype. Remember that this is encoding some information. Different genotypes we've seen in this course encode genetic information in different data structures. We've seen strings of floats. We've seen trees. Whenever we talked about genetic programming is that particular class of evolutionary algorithms that tends to encode genotypes in trees. Today we're going to look at genotypes that are encoded as graphs. A graph is made up of a series of nodes, the circles that you see over here, and edges that connect nodes together. That's a graph. The genotypes in the Sims work are particular types of graphs, nested, directed, multigraphs. So we'll unpack each of these three adjectives as we go. But for now, we're going to think of these genotypes as graphs. And the phenotypes, again, hopefully look pretty familiar to you. Strictly speaking, they're trees. There is some root node or root link, if we were to do this in PyroSim, and a series of links that are connected to parent links with rotational joints. Nothing out of the ordinary here. This particular mapping, the way that we're going to turn these graphs into trees is a recursive algorithm. We haven't gone into the details of exactly how this mapping happens yet, but you should be already able to see there's some visual hints here about the recursive nature of this mapping. How is it recursive? What does it mean to have a recursive genotype to phenotype map?
That the phenotype influences the genotype?
That the phenotype influences the genotype. That could be the case. It's not the case here. A good guess.
The graph has self-connections.
The graph has self-connections. We're going to walk through this G to P map in a moment, and as we do, we're going to walk through this graph. We're going to read out this blueprint in a recursive manner. Some of these links are going to bring us back to places we've been before. And as we walk along this graph, because it's a blueprint, is going to tell us how to build stuff. And as we build stuff, we're going to get things that have self-similar structure. What do I mean by self-similar structure? The recursiveness of this GDP mapping is going to tend to produce phenotypes that have certain features. They tend to look self-similar. What does that mean?
They look the same at different scales.
They look the same at different scales, right? So if we look at just this part of this phenotype, we get this little Y structure, and then if we zoom in a little bit more, we see that same Y structure again. Zoom in again, we see that Y structure again, and so on. This is self-similarity, or depending on the phenotype, it can be a fractal structure. You zoom into human physiology, or the physiology of most every organism, it is also self-similar. If you were to look at your own vasculature system at the whole body level, it looks like a tree, a whole bunch of big trunks pumping blood into smaller branches. If you zoom in on any part of your vasculature system, you see smaller and smaller branching all the way down to the molecular level. Self-similarity tends to be ubiquitous. It's everywhere in organism phenotypes. Let's just connect this back for a moment to our discussion about hyperneat and CPPNs, compositional pattern producing networks. You remember that evolutionary algorithm would paint patterns into a space of arbitrary dimension. That G2P map, the G2P mappings that was built into CPPNs, that mapping was designed to also produce self-similar structure or bias evolution towards symmetry, repetition, and so on. So the art of creating a good G2P map is that a random genotype, a random blueprint, doesn't necessarily produce a random collection of parts. It tends to produce phenotypes that have particular features. And typically, these maps are designed to produce typical features that we also see in nature, like, for example, symmetry. In our species, the left side of our bodies tend to look like the right side of our body. If you look inside us, you tend to see self-similar structures, and so on. Constructing a good G2P map is a very difficult thing to do. It's still a bit of a black magic, a black art, dark art. It's not so obvious to see how to do it. This particular one is complicated and seems to work well. One of the reasons why seems to be is that when you evolve populations of these things, populations of these genotypes, they tend to produce self-similar structure. Remember the video that you just saw? You actually saw, perhaps without realizing it, a lot of examples of phenotypes that had self-similar structure in it. Like what? What did you see in the video that looked, in retrospect, self-similar?
Crab.
The crab? How so?
It was symmetrical. It had two arms.
Absolutely. It was symmetrical, not necessarily self-similar. Self-similarity is not necessarily the same thing as symmetry. You can have something that's symmetric but not self-similar. You can have something that's self-similar but not symmetric. But yes, one of the things that this G2P mapping gives you, in addition to self-similarity, is symmetry. Actually, you can see it here. All three of these phenotypes are bilaterally symmetric. There was that kind of worm. You can go back and look at the video, and every segment of that worm was a little bit smaller than the segment in front of it. If you kept zooming in on pairs of those segments that made up that worm, you'd see the similar pattern at every scale. A big one followed by a slightly smaller one. Okay. All right. So let's have a look at how this works. Let's dive in now and look at some more detail of the genotype. As I just mentioned, the genotype is encoded as a graph. A graph is a series of nodes, and those nodes are connected with edges. In these particular genotypes, in these particular graphs, they are labeled. Every node and every edge has a bunch of numbers associated with it. That's the blueprint. That's the information. Okay. Let's have a look at some of the labels, some of the numbers that are attached to each and every node in the graph. There are 3 numbers attached that describe the body part dimensions. They describe the length and width and height of the block that should be built whenever we visit that node. You'll notice that these nodes have little names associated with them. This is meant to be a reminder to the reader that each node describes how to build a single link. If we want to build a single link, we need to know what the length and width and height of that rectangular link is. We'll come back to the joints in a moment. We'll come back to some of these nodes. We're going to have to jump around a little bit in the description of this genotype. Okay, so let's start to actually execute this G to P map. Let's imagine that this little node, or this little graph, is sitting inside a population of graphs. We have a population of these genotypes. We need to take each genotype and turn it into a robot. We need to turn it into a phenotype. How do we do that? We start by visiting the root node in the graph. There's one particular node, which is sort of arbitrarily denoted as the root node. We go there first, and inside that node, we find 3 numbers, length, width, and height. We build a link of exactly that specification. And now we leave this node, and we travel along all outgoing edges. Here we go. Okay. We would typically do this in parallel, but for the sake of this morning, let's do this one after the other. So we just built this particular link here using the information that's inside this node. We travel out along this link, or this edge, and edges also have a bunch of numbers associated with them. There's a bunch of labels attached to each and every edge in the graph. Those labels tend to be deltas. We just left this node, and we just created a node of a particular length, width, and height. So when we leave that node, we're going to take all of that information with us. We're going to carry that information in memory, and these numbers are going to tell us how to alter those numbers. There are 3 numbers associated with an edge that tell us how to change the scale. They tell us how to change the length, width, and height into some new length, width, and height, which we'll call L prime, W prime, and H prime. So far, so good? Okay. We talked about 3D orientation a few minutes ago. We're also going to take with us roll, pitch, and yaw, which perhaps at the beginning is just 0, 0, 0. There's another 3 numbers associated with this edge, a 4th, 5th, and 6th number that tell us how to change the orientation of that object. So we get a new roll, a new pitch, and a new yaw. So far, so good. There is a 7th, 8th, and 9th number that tell us how to change the 3D position of the center of the link that we just built. So far, so good? Okay, let's pull back a little bit and think about the intuition of this. We visit the root node, we build a link, we travel along edges, and as we do, these 9 numbers are changing. When we finish traversing the edge, we arrive back at some other node, or possibly the same node. When we arrive at that node, we have a length, width, and height. We have a roll, pitch, and yaw, and we have a new X, Y, and Z. Drop a link. We create a new link. So every time we visit a node, we create a new link. Everybody see that? So far, so good? Okay. Obviously, we need some joints as well. When we are sitting at a node, you'll notice that there's a bunch of joint parameters as well, but this is a little confusing. A node is telling us how to build a link, The node is also telling us how to build a joint. But a joint requires two links, right? How do we interpret this information? That this node in the genotype, in the blueprint, is telling us how to build a joint, but a joint that connects what to what? Do you think? Let's go back to the beginning of this G2P map. We started here at this node, and we just built a single link. There's information that's telling us how to build a joint. How do we build a joint? We don't even have two links yet. We only have one.
You just go to the end of that joint and then when you put the segment on, you connect it to the joint that you just built.
That's it. So this very first link that we build, we ignore the joint information. But as Emily said, when we leave a node, we know what we just built. We just built this one. We travel along the edge. And as we travel along the edge, we come back to this node or another node, and we build another link. So we just built a link. We remembered which one we just built. And now we're sitting at this node again. Now we take this information, which tells us how to connect the link we just made with the link we just made previously. Everybody see that? Okay, so we just built the trunk. We went along this left edge and we built this left branch. Pause. We just built this trunk. We simultaneously, at the same time we went along the left link, we also went along the right link. This link also has information about how to change size, orientation, and position. And those 9 numbers over here, when we come back here, tell us how to build a different link, the right branch. So we've visited this node 3 times, and we've traveled along 2 edges so far. So far, that's given us a phenotype that's made up of 1, 2, 3 links and 1, 2 joints. Everybody see how this is going? Okay, it's a lot of information to throw at you. I'll just pause for a moment. Questions? Anything that's confusing? Question?
Yeah, maybe we haven't gotten into it yet, but in the edge labels, have we spoken as what reflection is? Is that?
We haven't. We'll get there. There's some additional labels that we haven't talked about yet. We'll get there in a minute. Nate?
I'm just curious about what it means to traverse all of the edges from a single node. Like in these cases, they're all symmetrical. So it makes me wonder, like, is what you do on the left automatically mirrored on the right?
Absolutely not. It just in this simplified example, yes. But the deltas over here, the instructions about how to change size, orientation, and position, those 9 numbers don't have to be these 9 numbers over here don't need to be the negatives of these numbers over here. They could be 9 completely different numbers, which would produce a non-symmetric tree, but that tree would still be self-similar because this thing is still recursive. Everybody see that? Okay, good observation. Any other questions or observations before we push on? Yes?
All these creation happens before even simulating it?
Absolutely, right? So good point. So the genotype to phenotype mapping is just telling us how to construct the thing, the robot, in the simulation. We haven't even sent this thing to the physics engine yet. We just talked about creating URDF files. The physics engine needs the physics engine needs a blueprint. It needs information about what to build. It's a little confusing. This blueprint is building this thing, which in turn becomes a blueprint that's interpreted by the physics engine, right? We're going to take this thing in a moment, we haven't got there yet, and put it, or Sims is going to put it in his own physics engine. That reminds me of a detail I forgot to mention. Sims wrote this physics engine himself from scratch. You'll notice that there's some ray tracing, shadows, Are these necessary for simulate? An incredibly talented person. These are one of these human beings that was sort of ahead of their time. A lot of details in here that we don't necessarily need. This was the first physics engine ever made. Built by hand. I don't know about you, but I would love to see the code of this physics engine. Good luck. I've asked Carl a few times. He's pretty tight-lipped about it. If anyone manages to convince Carl to show us the code, I would love to hear from you. Okay, sorry, bit of a discursion there. Other questions, comments before we push on? Okay, let's talk a little bit about the rest of these labels. The rest of them are kind of optional. They're kind of bells and whistles. The most important ones are, what do we build? How do we create the joint? And how do we alter what we build as we travel along these edges? As long as you get that part, you're good. Okay. All right, let's talk about these additional labels, which again are sitting on each and every node. We know which two links to attach together. That's built into this G to P map. But how exactly do we build that joint? There's an additional integer here, which tells us which kind of joint to build. We could connect this link to this link with a hinge, if the genotype tells us to do that. Rotational joints that you are all now very familiar with. The genotype might instead tell us to connect those two links together with a ball and socket joint. Some of you have tried to simulate or approximate this now in your final project. What do you think joint limits means? Different physics engines have slightly different words for things. You're all experts on physics engines now. What does joint limits mean, do you think?
It's the farthest offset from the original position.
Absolutely, right? So if we're building a hinge joint, does it have wide limits or narrow limits? The genotype is again dictating some features now of the joint itself. Okay, let's talk about this one, recursive limit. This one is kind of interesting. There is a number, there's an integer sitting inside every node. It's an integer that's interpreted as the recursive limit. Let's go back and have a look at this genotype. When we first start interpreting this genotype, we visit this node. It tells us to build this trunk. We go out along both outgoing edges simultaneously, which means we come back to this node a second, then a third time, which causes us to build this and this node. For each of those second and third visits, we go back out another 2 times and another 2 times, which causes us to build this, then this, then this, then this. This is the recursive part, right? If we kept going, we would build a tree of infinite depth. We'd have a tree made up of infinite links, which of course would kill your physics engine. So like any recursive algorithm, which is exactly what we're looking at, this G to P map is a recursive algorithm. We need to give it some way to know how to stop. Every time we visit this node, whatever this recursive limit integer is, when we leave that node, we decrease that integer in the node by 1. If we ever arrive back at this node and RL equals 0, we stop the traversal of the graph. Okay. We've got this genotype, which is a graph. We've got a whole bunch of numbers labeling each node, and we've got a whole bunch of numbers labeling each edge. We haven't talked about simulation yet. We haven't talked about the brain of this robot yet, and we haven't talked about the evolution of these robots yet. Once evolution starts running, once we start evolving populations of these genotypes, what do you think happens to these numbers inside these graphs? What is evolution going to do to these genotypes?
Just tweak them.
Tweak them being what?
All the different parameters.
All the different parameters. A mutation can hit any label on any edge or any node. In addition, there are 2 additional mutational operators. There are 2 additional ways that the evolutionary algorithm can mutate these genotypes. Mutations can fiddle with all of these numbers, and it can also, what do you think those 2 additional mutation operators are?
Add a body part.
Yep.
Say add a node or add an
Absolutely. Not add a body part. A body part is part of the phenotype. Mutations don't act on phenotypes, they act on genotypes. A mutation can add or remove an edge or add or remove a node, which often has the result of adding, removing, and or modifying one or more body parts. That's the phenotypic impact of a mutation to the genotype. Yeah? What happens if a mutation, let's take this genotype right here. Imagine this genotype survives and produces an identical copy of itself, a child graph, and one and only one mutation event occurs, which is a mutation hits the recursive limit inside this single node, and it increases the value of this recursive limit. Let's test our intuition, or let's test our understanding of this G to P map. What's different between the child phenotype and this parent phenotype?
You're going to get another pair of branches added to the end of every branch at the outermost level.
Absolutely. In the child, the recursive limit is higher, which means when we translate the child's genotype into its phenotype, we're going to traverse through this graph recursively one more time or one more level, which is going to sprinkle, as you mentioned, a series of pairs of twigs on the end of this tree. We're going to get a slightly more arborized child compared to the parent. If the mutation instead had randomly decreased this recursive limit, the child would have less branches than the parent. Yeah? One way of building up your understanding of how this particular G to P map works, or any G to P map, is to try and visualize what happens if the genotype changes, what kinds of effects of phenotype could happen. Okay, let's play this game one more time. Let's take this parent genotype. It produces an identical child graph. And now there's a mutation to the change in scale on one of the edges. Let's imagine a mutation hits this edge, and it hits one of these three numbers. It alters the delta in scale. How does the child look different from the parent? This one's a little trickier. What's going to be different?
So it depends on what direction it changes in.
Okay.
But it's the children branches on, if it's just on one edge.
This edge. It's the mutation hit this edge.
Right. So let's say the delta, the delta is going to make it so that the branch is proportionally larger.
Okay.
You're going to end up with one side of the tree that's going to be heavier looking than the other because each branch is only different, is different by a different amount, if that makes sense.
You got all of it right except one little piece. You're absolutely right. Let's assume that the mutation increased the delta of the scale. It's telling this new branch to be a little bit bigger relative to the parent, to this link, than it was in the parent. You mentioned the left side of the tree.
It wouldn't be the left side, it would be each branch, the left one would be bigger.
Among every pair of branches, among this pair, the size of this link relative to this link is going to be different in the child than in the parent. And this branch is going to also be different in size compared to this one. This one is left unchanged. So a mutation has changed one number in the genotype, but that has affected more than one number or more than one part of the phenotype. This is a complicated genotype to phenotype mapping. Same goes for you. If you have an offspring and there's a mutation that hits one nucleotide in your genetic instructions, there's just one change in your set of genetic instructions compared to your offspring that is unlikely to affect just one and only one part of your offspring. G to P maps in nature and here don't work that way.
So I've got a question about the way the recursive nodes work. Would there be a way to, this is going to sound weird, would there be a way to have a variable number of recursive edges at each time the node is visited? So like, could you have it so that there's the option of either 3 branches or 2
Yes.
branches?
Great question. So imagine a phenotype in which the left side of the tree only has a depth of 3, but on the right side of the tree there's a depth of 4, for example. That's a particular phenotype. What would be a genotype that encodes that? Is there a genotype that encodes that? It's actually a very good question. Hard for this setup to do it, not impossible, but it can be done. We could also make a change to the genotype to phenotype mapping. I've explained to you how Carl came up with this G to P map, but we could make an alteration to this G to P map to make what you just said easier for evolution to do. How? How might we do it?
So would it be, instead of having it loop back, have 2 more nodes, like from the center node, and have those loop back in each other?
We could start to build more nodes and more edges to approximate this depth 3, depth 4 tree. It'll work, but it's a little bit inelegant. We're adding a lot of stuff to the genotype. Can we make just a slight change to how this whole works, that this would still produce a depth 3, depth 4 tree?
Could you keep track of the recursive limit on the edges instead?
Bingo. Move this label, this integer, from the nodes to the edges. If we have an RL label of 3 sitting here, actually no, that's still not going to work. That means the left pair of branches is always going to be shorter than this one, but I think you're on the right track. That's the way we think about G to P maps, exactly, right? We made a change to the overall G to P map. We moved this label from a node to an edge, and that alters the kinds of phenotypes we tend to get with these genotypes.
I think it might be fun to have edges that have a probability of being found.
Okay.
That sort of gets in it. Would be
Yep.
different.
Absolutely, right? We could make this a probabilistic recursive G to P map. All of these numbers represent probabilities of things happening more often or less often than others. Great. Lots of ways we could do that. What would you tend to see in the phenotypes, do you think, if you had some probabilities incorporated in the genotype? What would happen to the phenotypes in general?
See more variation in their designs.
Absolutely, right? So in this tree, every single branch has exactly the same depth, right? Uniform. If it was probabilistic, that would no longer be true. That might make it easier for evolution to evolve robots that do whatever we want them to do, or maybe harder for evolution to evolve robots to do whatever we want them to do. Another issue we haven't touched yet about G to P maps is, do they make it easier on evolution or harder on evolution to produce whatever we want evolution to produce? Another open problem in the field. Nobody knows. You come up with yet a different G to P map. Is it going to make things easier or harder? No one knows. Other questions? Okay, let's keep going. Somebody mentioned the reflection label down here. It doesn't say it here. This is a binary label. It's either 0 or 1. As we're going through here and we're executing these deltas, the way that we build things, the reflection can sort of flip things around as we continue building. I think that Carl threw it in, although he didn't say explicitly in the paper. I think he threw it in to make it more likely for phenotypes to have symmetry. Hard to say. It's not too important for our purposes. What about this last one? Terminal only flag. It's called a flag. So for computer scientists, that's a strong signal that this label is a binary number. What does it do? What does it do? And why did Carl throw it in, do you think? It's a label that's sitting on edges. Every edge has one of these TO flags that's either 0 or 1 sitting on that edge.
Only thing I can think of is like letting the node know not to build a joint.
It lets not the node, it's the thing that's constructing the phenotype, our program instruction counter, whatever, that's sitting on a particular node or edge. It's telling it, go along this edge or don't go along this outgoing edge.
It'd be really useful to have like a claw tip at the end of your arm. You could say, oh, leg segment, leg segment, leg segment, and when you run out, put a claw.
Absolutely. Terminal only flag here. So if the terminal only flag is set to 1, that's the edge saying, stay away from me. Do not travel along me. Build whatever you need to build. And when you're sitting in a node and you're considering traversing along me, you're only allowed to traverse along me when the node's recursive limit is 0. Once you've reached the recursive limit of building something, now you're allowed to follow the edge and build whatever you arrive at on the other side of the edge. Carl probably threw it in thinking that for some of these creatures, it would be good to have a unique structure at the end of some of these appendages. A claw, a head, a foot, a hand, who knows? Okay, all good? Okay. All right, let's again test our understanding of this G to P map. I've left on the right-hand side of the slide for you all the labels, all the node labels and all the edge labels. I want you to take a minute. You can do this yourself or turn to a neighbor and talk it over. Pick one of these robots and think about, can you write down a genotype, maybe just the nodes and edges, that's likely to produce that phenotype? I'll give you 2 minutes to think on that. See if you can write down some of the genotypes for some of these phenotypes, and then we'll see what you came up with. Pen and paper might be useful here. Some are easier than others. Okay, let's start with the 10,000-foot view here. Which of these 12 phenotypes do you think has the simplest genotype? Simple meaning the fewest number of nodes and edges.
8.
Why 8?
It's like the snake, it's linear and cats loop back on. You only need one edge and one node to loop on itself.
It's possibly that you could get away with one node, one edge for 8. Maybe, although I see a little bit of structure in this robot that says maybe not.
I think it's 10.
What's that?
I think 10 is the easiest.
10 is the easiest? Okay, I hear a vote for 10. Emily? 7. 8, if you squint, you can sort of see there's 1, 2, 3, and then 1, 2, 3, and then 1, 2, 3. There's a little, it looks like there's a couple of kinks in here. Hard to say. My vote is with 7 as well. We have one node. We imagine that there's one node that tells us how to make the head, and one outgoing edge. And I'm not going to write down all the labels for this one, but one that says to change decrease the length, decrease the height, and decrease the width by a little bit. Build something a little bit smaller, then a little smaller, and a little smaller, and a little smaller. I would bet that you could probably construct by hand a genotype and fill in all the node labels and the edge labels here that would produce something that looks quite a bit like this. Anyone tackle any other robots? What might be a genotype for number 6? How many nodes and edges?
For 6, I thought you'd have, like starting at the bottom center cube, that would be one node, and that loops on itself, like to make the 3 center ones. And then it has,
I'll call this segment.
yeah, and then there's another node that does little wings, and from each center segment, there's 2 edges that could go to that.
We'll make a node called wing node. I think you're right. This, something like this, would probably build something that looks like this. Everybody with me so far? Okay. Okay, so let's go back for a moment and talk about the actual name for these genotypes. Now we're ready to unpack this term. We now know why these genotypes are graphs. They're made up of nodes and edges. It's a directed graph, meaning that all of the edges have an arrow associated with them. They have a direction. We know when we're at a node, we know which of the edges are outgoing edges, and for that node, we also know which are the incoming edges. So we've got a directed graph on our hands. If you've ever taken a complex systems course or a math course, you've probably come across directed graphs before. What's a multigraph? You can have multiple edges between the same pair of nodes. We now see why it kind of makes sense to have a multigraph. It makes sense when you're going to create different versions of things to not duplicate the genetic information. It's in there, but the deltas alter how you build those things.
Does this mean that it's modular? Like you can just do the head in one side and then the body in the other?
Absolutely. This GDP map has been around for a very long time. Many people have used it, used versions of it, because among other things, it's modular. A mutation to any label of any node or any edge may impact just one part of the body, maybe just the head, and leaves everything else alone. Or alternatively, a mutation to a node might, or to a label might affect many or all parts. Evolution can decide how modular or how non-modular to make these genotypes. We actually dedicated an entire lecture to an evolutionary algorithm that was designed to be able to increase or decrease the modularity of the phenotype. Same thing here, but in a very different way. Yep, good observation. Okay, we've unpacked 2 of the adjectives here, directed and multi. What about nested? I've deliberately hidden this last one from you. These genotypes, as you're going to see in a moment, are also nested. What do you think that means, first of all, just in general?
There can be like different graphs inside each other.
Inside every node of these graphs, there is another graph. Inside this node is a series of nodes and edges, which we haven't seen yet. What do you think those embedded or nested nodes and edges do? They're in the genotype. What haven't we discussed yet? Absolutely. Okay. Here's a genotype on the left, which looks more or less like exactly this. And you'll notice this is a nested graph. There are nodes and edges inside individual nodes. Each nested node, so we're going to talk about nested nodes and then just nodes. Each nested node dictates how to construct part of the neural controller for this robot. All we've seen so far is how the genotype genetically encodes the body. Now we're seeing how the genotype also encodes the brain along with the body. If you thought this GDP map wasn't complicated enough, Here we go. Okay, so each nested node dictates some part of the body, and each nested edge, these thin arrows in this picture, denote how to build some other part of the body. We've got nested nodes and nested edges. How do you think these two things collectively dictate how to construct the neural controller of the robot? Strong hint, nested nodes, nested edges, telling you how to build the brain. Which does which? We've drawn and seen many neural network controllers in this class, and they tend to be drawn as graphs, as nodes and edges. Each nested node has information inside. It also has labels that tell the G2P map how to build a neuron. And every edge, nested edge, tells the G2P map how to build a synapse. Okay. You'll notice things like wave, absolute, sawtooth, wave. We're visualizing here some of the labels of the nested nodes. These names are meant to be strong hints again about which feature of neurons these nested nodes are encoding. Sorry? Not behavior. We're just talking about neurons. Yeah, wave. Yeah, absolutely, a waveform, but not about the behavior. The activation functions. Each nested node here has many labels associated with it, and their details we're not going to get into. One of those features is what is the activation function inside the neuron that's constructed. So far, so good? Okay. Nested edges that specify how to build a synapse. These nested edges have one and only one label attached to them. What is it, do you think? A nested edge tells the G2P map how to construct a synapse inside the robot, and there's one number associated with that nested edge. What is that number encoding?
Synapse weight.
The synaptic weight. In this class, the only feature we've talked about for synapses is their weights. Yeah, that's it.
What is IFP or ITP again?
I don't remember. Sims came up with a very large number of activation functions. ITP is interpolate. Not a very common activation function. What's he doing here? He threw in every activation function he could possibly think of and just sort of is telling evolution, here you go. If any of these are useful when constructing the brains of these robots, knock yourself out. Okay, we've got 2 minutes left, so let's get over the last intellectual hump of understanding this G2P map. Two more I want to talk about. So we see there are some of the labels inside these nodes are J0 and J1. This is a little confusing. These are joint sensor neurons. Some of these embedded nodes are telling the G2P map how to build hidden neurons. Some of these nodes, like these two, are telling the G2P map how to build sensor neurons. These are sensor neurons that, as the name implies, are attached to the joints. And these joint sensor neurons detect and report out the angle of the joint. These are proprioceptive sensors. We've seen these called angle sensors. We've seen these called proprioceptive sensors. For Sims, these were called joint sensors. There are also two motor neurons here. These motor neurons are sending out motor commands to the two effectors. Again, another synonym for motors in robotics. This node down here, this is the wing node. This node is visited 4 times. 1, 2, 3, 4 times when we're traversing this genotype, which means we visit this node 4 times. What can you tell me about the neural tissue or the bits of neural network controller in these 4 wings? If we were to crack open any one of these wings, what would we find inside? Do you think? Would we find no neural material, some neural material? What would it look like? What would we see inside?
It looked like the right half of that diagram. You have inputs for the joint position, outputs for the motor position, and those would be duplicated for each of the wings.
They'd be duplicated. Inside each of these 4 wings, you'd find this 4-neuron neural circuit. This is a peripheral nervous system. Bits of the brain that are in different parts of the body. We have neurons and synapses in our arms and legs and in our spine. That's your, taken all together, that's your peripheral nervous system. And then we got a whole bunch of neurons and synapses centralized in one part of our body. That's the central nervous system. We'll end here today. These robots can also have PNS and CNS, which we'll talk about next time. You have a quiz due tonight. You're working on your final project. See you Thursday.